Today, I'm going to walk you through the setup for your Photosynthesis and Cellular Respiration Lab. So I've got my materials here. I have, as you can see on your instructions, my pond plant. Okay. I have my 3 test tubes with the caps already screwed on. I have a small beaker. I have 2 large beakers. And I have a straw, and then in this flask I have some bromothymol blue, which is our indicator that will tell us if there is oxygen or carbon dioxide present. So, step 1 is going to tell me to get my large beaker, right here with the straw in it, and I'm going to measure out 60 milliliters of my solution. Now, this is not going to be super exact because it only shows me the 50 and then the 100. So if you want to use a graduated cylinder for this, you can, but it doesn't have to be perfect. So I'm just going to pour mine in till it's a little bit above the 50, just about there. Now I'm going to use my straw to blow bubbles into my bromothymol blue solution. So I am exhaling my carbon dioxide into the solution to change its colors. Now please don't share straws. Decide which person on your table is going to use the straw and only let them use it. Okay? Okay, I can see a pretty good color change there with just one deep exhale. If I want to keep going, I can, but it probably won't show a whole lot more than that. I think that's good. All that you want to be able to do is see the color difference, which you can clearly see. When you are blowing bubbles, try to make sure that you're not blowing so hard that the solution just goes everywhere and makes a mess, because you will have to clean it up. Okay, now that I've got my solution, I'm going to, make sure I'm going in order with your papers, pour 20 milliliters of the solution into each of these test tubes. That's why I have my small beaker. So, in my small beaker, I'm going to measure out 20 milliliters. Okay, up to there. It won't be quite enough to fill up each test tube, and that's okay. Again, I'm not being super precise on this. I'm just getting close to 20 milliliters. Perfect. If you have any extra left in your beaker, then just go ahead and rinse it out in your sink. Okay, I am now done with those. So, step number 2 tells us to put a piece of a plant in test tube 1 and put the cap on. So up at the front of your lab, you should have something to get this out with. My jar's a little bit small. Okay, I'm just going to get a piece of this moss about like this. This is plenty. Okay. I'm going to put it in test tube number 1. Put it all the way down in the solution. Okay, I'm going to go ahead and put my cap back on. There's test tube 1. Test tube 2, I am going to put a piece of plant in that one as well. Okay, about the same piece. This is plenty. You can even do it with a little bit less than that, and I bet you'll be just fine. And test tube 3, I'm going to leave without a plant. So I'm just simply going to place my cap back on. Okay, next step. I have got to put foil on test tube 2 because we're going to test test tube 1 and 3 in the light and test tube 2 in the dark. And we just want to make sure that there isn't any light showing through. So I'm going to take off a piece of foil, oh so ungracefully, and I'm just going to wrap my test tube 2. Okay, so that there isn't any light that gets into it. All right, test tubes 1 and 3. I'm going to put in this beaker. These are going to go outside so that they can photosynthesize in the light, and I'm going to need to label this with my group's name. So make sure you put everyone in your group's name, or at least a few of you, so that you can identify it. This I'm going to set aside and I'm going to take outside. Now I'm going to need to use my other beaker that's been rinsed out. And I'm going to put test tube number 2 in this one. And I'm going to open up my cupboard underneath my station and place it down there. At the end of class, we'll gather all of the test tubes together and see if we can identify any color changes in the bromothymol blue solution.